Okay, so yeah, I think it's time. So good morning, good evening, good afternoon, everyone, depending on where you are. So today's a pleasure to in this another edition of eoPERCS to have Bill Gearty with us. He's going to be talking about body mass constraints on aquatic invasion in tetrapods. So before we go to the actual talk, as some of you know, we're going to go through today's seminar format. We have this 5 minutes welcoming announcements, followed by Will's talk, and then we're gonna have a moderated Q&A. After that, we're gonna have tea time with Will, and in which he talk a bit more about research and other stuff. And here's the first reminder that you can send questions to Will via our questions host by the chat to our questions host of today's Natasha. Um, so just a little bit of housekeeping before we go to the talk. eoPERCS values the participation of everyone interested in the neurosciences. So remember to abide by our code of conduct. If you haven't done so, please go to our website and do it. You should not be able to unmute yourself, but if you do for any reason, please remember to keep yourself muted during the duration of the talk. Another reminder that you can send your questions to our questions host by the chat, but you can also raise your hand using one of the options down there in Zoom. I think it's which comes, I never remember. Reactions. Yeah, there's a reactions button. Usually you can raise your hands and we can unmute you and you can ask your questions directly to Will. We also have captions built into Zoom. You can hide or show them using the little CC button down there as well. Please remember to nominate yourself through our website. Nominate yourself. Nominate early career researchers through our website. Anyone that you know that is an early career researcher that works on many career fields. You can also nominate yourself if you want to. You're going to see a link to that in the chat soon. And finally, we have we have a demographic survey for weekly feedback information. It's anonymous, it's optional, but we encourage people to do it, even if you have done it before. So finally, I would like to introduce Will. He did a bachelor's in geology and geophysics at Yale University, followed by a PhD in geological science at Stanford University, and is now finishing a postdoc at University of Nebraska-Lincoln, and we'll be starting soon in September, so in a few days, a new postdoc at the American Museum of Natural History. So with that, we would like to invite you to start your talk, and you can take over the screen share.
Great. Thank you so much, Pedro. And thank you to the committee for inviting me, and thank you to all of you for coming today. Today I'm going to be talking about some work I did back in my PhD. I would have made a different talk, but as Pedro said, I'm in the middle of moving, so I didn't really have time to put together a brand new talk. So apologies to anyone who's seen some of this before, but hopefully you still enjoy it. So today I'm going to be talking about 2 chapters of my PhD dissertation, looking at how body mass constraints, or how there were body mass constraints on aquatic invasions within tetrapods over the past 300 million years. And so about 300 million years ago, 350 million years ago, tetrapods, or at least the ancestors of them, invaded the land, and since then have diversified into all the clades that we know today: amphibians, snakes, mammals, birds, etc. However, during this evolutionary history, there have also been a number of reinvasions of the ocean. You can see here most of the ones that we know about 30, 40 odd reinvasions of the ocean within all of these different clades. And all of this has resulted in a bit of diversification. You can see here recent estimates of that diversity over time, split up by different clades. However, this really pales in comparison to the diversity that we know of marine invertebrates over this time period. And it also pales in comparison, interestingly, to the diversity of non-marine tetrapods that we have records of. And this is actually an underestimate of the non-marine diversity, we suppose, because we have less recorded terrestrial rocks than marine rocks. And so one of the questions I'm really interested in is, why do we have this depauperate marine diversity of tetrapods compared to their non-marine relatives? And really, I think it comes down to the fact that we have many different chemical and physical properties in the ocean versus on land. Things like varying amounts of oxygen availability, differences in salinity, differences in buoyancy, the amount of heat loss you experience, and also the viscosity of the fluid around you, that being air versus water. And it's no surprise probably that lots of tetrapods enjoy going in water from time to time. Here's just a smattering of images and videos of tetrapods enjoying their time in water. But ultimately, permanent life in the ocean is really hard, especially when you've adapted to living on land. Of course, it's not that hard for things that have adapted to living in the ocean to begin with. But once you've adapted to living on land, it's very hard. And that's because you now have to deal with a different salt balance, different ways of acquiring food, thermoregulating differently, reproducing differently, locomoting differently, and you have to also worry about dehydration. But the great thing is that a lot of these problems are actually overcome by body size. As you get larger, you can more easily thermoregulate your body in the water. You also can acquire more food, you can move faster, and so all of these things are actually overcome by increasing your body size. And so today I'm going to talk about how I've looked at this in mammals and crocodiles. So first, in mammals, here's the four groups of at least modern aquatic marine mammals. You have pinnipeds, including your seals and sea lions, Sirenia, including your dugongs and manatees, Cetacea, including your whales and dolphins, and then Lutrinae, including your otters. And just looking at these examples right now, you can see that all of them are on average bigger than their terrestrial counterparts. Seals and sea lions tend to be larger than things like red pandas and other caniforms. Dugongs and manatees tend to be larger than this hyrax, their Afrotherian relatives. Of course, there's also elephants in that group, but at least on average, they're larger than these Afrotherians. Dolphins and whales obviously are much larger on average than their artiodactyl relatives. And actually, the aquatic and marine otters tend to be much larger than their terrestrial relatives like stoats, shown here. And so this has been well known that they're quite a bit larger than their terrestrial relatives. But the question comes down to, well, earlier I presented the fact that there's actually a lot of different constraints, physical and chemical constraints, on why you might want to get larger. And so here I'm interested in can we actually pin down exactly which of those constraints, which of those evolutionary, physical, or chemical constraints drove this evolutionary change in body size? And so there's kind of two steps to go about testing this. First, you actually have to test that the body size, the evolution of these larger body sizes was actually an adaptation. And so you can do that using model fitting. Here I'm showing random versus adaptive evolution models. And on the left here is our random evolution model, also kind of often known as the drunk man's walk or Brownian motion model. You can see here that in this case, each step that you take through time on the x-axis is completely random in both amount and direction. And so here you can see this drunk man kind of walking down the street and he's kind of swaying back and forth. Whereas in adaptive evolution, you're evolving towards some optimal trait, some optimal phenotype. And so generally you start at some unoptimal trait or phenotype and you move towards that optimal phenotype over time. Now, both of these processes can actually be modeled by a single equation here, and that's called the Ornstein-Uhlenbeck process. You can see there's kind of two major parts here. So the amount of change that you expect, dxt, over time is related to both the amount of selection occurring, and that is related to what the optimal trait is, and also what your trait is at any given time, the strength of selection and the amount of time that's passed. And it's also related to some amount of random drift that's occurring, and that there's some intensity to that given by the sigma parameter, and there's also just kind of white noise occurring. And so together, these two can give us both of those processes I showed before. If you only have the second component, that white noise random drift component, you get a model that looks like this Brownian motion model. And you can see here that even though there are, where these dots are, these are speciation events. So even though they're speciation events, they're all kind of within the same Brownian motion process, and they're all kind of in the same realm of phenotypes. And you can see there is a little bit of a trend, perhaps, but that's actually expected in some cases in a Brownian motion model. Whereas if you compare that to an Ornstein-Uhlenbeck process that has both of these components, you see that there's actually evolution towards the optimal trait phenotype up here at larger body sizes. And then despite having these speciation events, they all kind of hover around that. And so that's kind of key about this part here, this theta minus x, is as you get farther away, you get pulled towards that optimal value faster. And as you get closer, you kind of enter a stasis mode, if you will. So that's the step one. So step one is we have to actually test whether these animals have been adapting, whether this body size increase is adaptive. Step two is then testing between those hypotheses that I presented before, or those driving factors. And so we can do this by looking at the parameters that you get from these fit models. One of those is called the phylogenetic half-life. You can see that here. That's ln2 over the alpha parameter, which is the strength of selection. And this tells us basically the time in millions of years that it takes to move halfway to that optimal trait value. And you can also think of this as kind of the rate of directional evolution. There's also stationary variance, which is this equation here, this formula here. And that's the expected amount of variation once you get to that equilibrium state. And so that's kind of your rate of random evolution. And so we can use both of these and the expectations of all of those different hypotheses to test between them once we fit our models to the data. And so coincidentally, 3 of those 4 different mechanisms that I mentioned before all actually predict that you would have an increase in your stationary variance. So basically thinking about habitat area, if you have more habitat area, you can potentially get bigger and eat more things because you have more habitat to explore, but you could also still be smaller. So once you get to some equilibrium value, you expect that actually the range of body sizes you expect will actually be larger than it was on land because you now have a larger habitat to explore, but you could still be smaller. The same thing goes with protein availability and also neutral buoyancy. All these things allow you to get larger, but don't prohibit you from being smaller. On the other hand, thermoregulation actually forces you to get larger because you're losing much more heat by living in the water. And so you have to counteract that by getting larger. If you stay small, you'll actually lose so much heat that you'll eventually die. So you have to get larger to counteract that, to maintain a surface area to volume ratio that allows you to maintain your internal temperature, especially since we're talking about warm-blooded mammals here. And so obviously there'd be a problem if we get some results down here. We wouldn't know which one of these to pick. But if it is up here, only this one matches the results. Anyways, I'll leave that for the results section. I went about collecting lots of body masses of mammals. In this case, I collected lots of living mammal species. I also collected fossil mammal species body masses. I won't be presenting that today, but the results are basically the same. I also had to code all these species for whether they're terrestrial or aquatic. In this case, aquatic includes freshwater species as well, just because of to increase the sample sizes for some of these, especially for Aphrotheria. If you only do marine things, then you get quite a small sample size for the marine group. And then I also used a phylogeny. So if you remember back in the equation, you have to account for the amount of time that's passed between these different species. And one really useful way to do that is with a time-scaled phylogeny. And in this case, basically, the amount of branch lengths that occur between any 2 species is the amount of time that's passed in terms of evolution between those 2 species. And so we can use the phylogeny to account for all of that between all of the different species. In this case, I did the analyses separate for Aphrotheria, Artiodactyl, and Caniforms. But you could theoretically do it with the entire tree. This was just to save computational time. But it should get the same result. Um, okay. And so then you go about fitting these models I mentioned before. You take all of the data. In this case, there's some uncertainty with the super tree, so you take 100 different versions of it to account for that uncertainty. You also, of course, have all these body masses and habitat codings. You combine that with specified models that you want to test between. In this case, we're testing between random evolution models and adaptive evolution models. And within each of those sets of models, we're testing everything evolving together, and we're also testing aquatic things evolving differently from non-aquatic things. So in this case, we should have 4 different models, and then there's some sub-models in each of those. You run this all through a software package called eoE, which is in R. I think there's actually been quite a bit of development on it in the past few years. Back when I used it, it was a little hard to use, but I think now it's much more user friendly. Anywho, I digress. From this eoE analysis, you then get a bunch of parameter estimates, including the optimal values that it estimates, those phylogenic half-lives that I mentioned earlier, and also the stationary variance. In addition to that, for each model, you get an AIC value, and you can calculate AIC weights from that in order to estimate the relative goodness of fit of the different models to one another. Okay, and so here we have our model fitting results. I've whited out the table just to walk you through it first. So each row in this case is one of the 4 groups that I tested it on. And then each column is one of the 7, in this case, models that I tested. In these first 2 columns, there's no adaptation occurring. In this one, there's no adaptation occurring, and they're all not adapting together. And in this case, there's kind of separate sigma terms, if you remember that variance term earlier in the equation, for the aquatic and terrestrial thing. Here, everything is adapting the same way, this OU1 model. And then all 4 of these represent kind of different levels of terrestrial and aquatic mammals adapting differently. And so if we are, if there is adaptation occurring, and that adaptation is different for the terrestrial and aquatic mammals, we would hope to see the most support in these columns here. And so you'll see that at least for the first 3 rows here, that's in fact what we see. We see lots of strong support for these OUM models, where terrestrial and aquatic mammals are adapting differently. Down here in mustaloids, which includes the otters and stoats and things, we actually have the most support for the random evolution models, although there is kind of a smattering of support for adaptive models as well. So there's maybe less support for these kind of models here, but also maybe indicating there's some support for them. Okay, and so 3 of these groups seem to be adapting differently upon entering the ocean. It should also be mentioned, just as an aside, that the otters are the most recent invasion of the ocean in mammals. And so it's also possible that we're not able to detect the adaptation occurring because there hasn't been enough history. If we look at those parameters that we're estimating, you'll see here that in all of these cases, the aquatic parameters are significantly larger than the terrestrial optimal parameters, by several orders of magnitude in some cases. You'll also see that for those 3 groups that had strong support for adaptation in the different groups, that they all kind of overlap at the same body size, which is about 500 to 800 kilograms. You'll also see that I didn't mention this before, but I separated out the baleen whales because obviously they're much larger than the other whales and dolphins. And so I wanted to treat them as a separate group, which always was a better fit than not treating them as a group. You'll see, of course, they have a much larger optimum here than the other aquatic cetaceans, or the other cetaceans. Okay. We also get that phylogenetic half-life parameter. Remember, this is the time it takes to get halfway towards the optimal trait value. And so a larger half-life means you're evolving slower, and a smaller half-life means you're evolving faster. And so in this case, we see that almost all of these groups are evolving faster. They're taking less time to get to that optimum, which makes sense. If this is a really important constraint on body size evolution, you'd want to evolve very quickly to get to that larger size in order to be able to survive in the ocean. Okay. And then we also see a decrease in the stationary variance. So there's less variance around the body sizes that are viable in these aquatic groups. And so if we come back to our chart of potential hypotheses, potential mechanisms, and drivers, you see that actually our results indicate this area up here, and that goes along with thermoregulation as a potential mechanism. But I didn't want to stop there. Sorry, my dog was moving. I didn't want to stop there. I wanted to go a step further and see, well, can we actually predict the sizes that they evolve to based on thermoregulatory mechanisms? And so I built an empirical model based on data from seals. And so in this case, we have feeding data on seals where you can see based on their mass, how much they eat per unit time in this case. And so we can put that right here, this yellow line. And of course, your feeding rate has to increase as you get larger in order to sustain your metabolism. But your metabolism also increases as you get larger. And so we have that green line here. You would want, at the very least on land, for your feeding rate to always be larger than your basal metabolism, so that you have some excess energy. In water, however, you now have another mechanism that's removing energy from your equilibrium, and that's heat loss. And in this case, I've treated these seals in this case as kind of an insulated pipe that you can then calculate based on the size of that pipe and based on some estimates of how thick blubber is, how much heat they'd be losing at particular body sizes. And so in this case, you can see that line is here in blue. And so again, in order to survive, and actually you want a surplus of energy after you account for all of these in order to reproduce and things like that, you need for the amount of energy left after feeding and taking into account basal metabolism heat loss to be greater than or equal to 0, right? And so really ultimately this section in here, a chunk of this section in here, this triangle, is what's viable for these organisms, these mammals. And so here's our raw data plotted as body mass histograms on the x-axis. And you can also see those HOWI parameter estimates up here for the different groups. Remember, here this model is based on the seals, so this dark blue. And here's the model, and so that's basically F minus M minus H, feeding minus metabolism minus heat loss. And you see that the excess energy that you get corresponds quite nicely to the observed body masses of seals and sea lions in the model. So, in conclusion, 3 out of 4 of these mammal groups that are living in aquatic environments today have larger optimal body sizes than their terrestrial counterparts. And as I said, I didn't show it today, but if you look at the fossil record, you see very similar results. All 3 of these groups are attracted to about the same optimal body size, about 500 to 800 kilograms. And this is in conjunction with increased selection and decreased variation, which excludes most hypotheses or mechanisms except for thermoregulation. And so I went ahead and built an empirical model that supports thermoregulation as the main driver of these body size increases, and observed that also the feeding rate limits their maximum size, which causes more constraint on size in water than on land. So because they still have the same feeding rate constraints as they do on land, they're still limited in size, and that causes this decreased variation. Of course, there's these baleen whales that get to much larger sizes, and so there must be some mechanism, such as engulfment feeding, ram feeding, things like that, filtration feeding. These things must be increasing their potential feeding rate at these larger sizes that allows them to have much larger sizes than even these other marine mammals. Okay. So moving forward, I also then wanted to look at the same question in crocodiles. And as you see here, crocodiles in the present are kind of all the same. They mostly live on land or in semi-aquatic environments. They mostly look like this, but back in the fossil record, they're quite diverse and disparate. Here, I'm showing on the left a number of different reconstructions of terrestrial crocs, and on the right, a number of different reconstructions of marine crocodiles. And you can see they cover quite a wide array of morphology and body size. And so the question here is, do ectotherms exhibit the same adaptation as endotherms? And is it for the same reason? So again, we can collect lots of body size data. In this case, we have to get it from fossils as opposed to from body mass measurements of modern mammals from the previous chapter. So in this case, I measured skulls and femurs from the primary literature, from photographs and things like that, and then use scaling equations to get mass estimates for those species. In many cases, I had multiple elements for individual specimens, and so I was able to get multiple mass estimates for each species and each specimen. And then I used various sources to code these species as either being terrestrial, semi-aquatic, or marine. In this case, I did separate semi-aquatic and marine because I did enough data to support those different categories. And that came from numerous different sources. And then, actually, this is wrong. Then I used the paleobiology database to get data on when these specimens occurred in the fossil record, and use that to then date fossil phylogeny. And so in this case, the phylogeny has over 300 species in it, and more than about 300 of those species are extinct, representing quite a bit of the diversity of crocodiles through time. And so basically in this case, the limiting factor is the size data availability for these species. In many cases, these species are included in this phylogeny based on individual teeth or part of a snout or things like that. And so you can't really get mass estimates from those. And so in this case, you're able to get habitat and range data for almost all of these species, but not size data. And so that is somewhat of a limiting factor. I timescaled the super tree using kind of a novel characterless tip dating method in Mr. Bayes. And you can see here that there's kind of three broad marine invasions. One in this Tethyseuchia group, another in this Thalatoseuchia group back in the Jurassic, and then there's kind of a smattering of marine invasions up here in Crocodilia, which we're kind of considering one invasion, but you can see there's kind of two main ones here. One thing I find really interesting is that this Tethyseuchia group actually makes it fairly untouched through the Cretaceous Paleogene mass extinction, and they continue for quite some time until they probably get outcompeted by cetaceans. But I don't think anyone's actually really looked at that. Okay, this is just a summary of the body mass evolution through time. This doesn't use any sort of modeling yet, but this is just showing you along the phylogeny how body masses have been changing using just a normal Brownian motion model. And so you can see that just looking at the data, that these marine crocodiles in blue have been evolving from much larger than average ancestors to begin with, and then sometimes even evolve to larger sizes after that point. And you can also see that the semi-aquatic and terrestrial groups here are fairly similar across most of the evolutionary history. So I did a very similar model fitting approach in this case, again, using a time-scaled phylogeny, in this case, 500 different versions of it to account for all the uncertainty involved time scaling and also resolution of polytomies using that data I talked about that I collected. And then in this case, I did quite a few different models. I think it was over 20 different models, if I remember correctly. But again, we have our random evolution models, we have our adaptive evolution models, and then within those categories of models, we have everything evolving together. We have marine and non-marine things evolving differently. We have aquatic and non-aquatic things evolving differently. So aquatic here includes marine and semi-aquatic. Non-aquatic includes just terrestrial. We have all three of the habitats evolving differently. And then we even have all three habitats evolving differently with a clade effect included. So where the crocodiles might be evolving differently from the Tethyseuchians, et cetera. Again, we get all of those same parameters and model support out of it. And so we can look at the results here. And so in this case, because there are so many more models, I can't have columns and rows for all of them. In this case, I've split it out by those categories that I mentioned earlier of how we're grouping the things together. And then all the different models are color coded by gray here for random evolution and blue for adaptive evolution. And so what you can see is that the models with the most weight with these stars here, and you can see have the highest box plots here, are this OUMVA model, which is an adaptive evolution model, and where you have marine versus non-marine or three habitats differently, or even with a clade effect. So there's kind of split support for these three different models, all of which indicate that marine things are evolving differently from the other groups, and all of which indicate there might be some, or at least this one also indicating there might be some clade effect going on. So in order to account for this variation here across these different model fits, I averaged across these three models to get the results I'm about to show you. And so again, in this group, we see that they also get bigger, just like the mammals. The semi-aquatic species groups tend to be about the same size as their terrestrial counterparts, and the marine groups tend to get much larger by multiple orders of magnitude. And this size actually corresponds quite nicely with this about the same size in the mammals, about that 500 to 800 kilograms. That's really cool. We also see that they have stronger selection, faster rate of evolution, marginally in this case, but it is significant. But it's not necessarily as much of a change as in the mammals. And we also see that they have less variance than the other categories, the other habitat categories. And so coming back to our mechanisms, and here you can see that this is basically the same as what we had for mammals, except in this case I've included lung volume, and I'll get to that in a second. But in this case, we have the exact same set of boxes here as we did for mammals, indicating perhaps a similar mechanism driving this body size evolution. And so, like I said here before, it could be thermoregulation like with the mammals, but it could also be lung volume, which has long been held by herpetologists and paleontologists as what's driven larger size in these crocodiles. Now, at face value, lung volume seems like a reasonable mechanism to drive this body size increase. Obviously, it limits how long these crocodiles could hold their breath and dive to feed in the ocean. And it does, in fact, increase slightly as crocodiles get larger. And we can measure. We have data on modern crocodiles and how their lung volume compares to their body mass. We also have data on the rate of oxygen consumption based on body mass. And so you can calculate basically their diving capacity in minutes roughly based on their lung volume. And it's also possible that they are storing some oxygen. Mammals do this quite often, marine mammals store lots of oxygen in their blood. But I imagine that also scales with lung volume. And so it might kind of move the y-intercept here, but it shouldn't change the slope much. And so you can see it does increase a little bit their diving capacity as they get larger, but it only goes from about 30 minutes to 100 minutes. So you're maybe doubling, tripling your diving capacity, and it certainly isn't limiting you in any of these body masses, because even at the smallest body mass, the smallest crocodile could dive for at least 30 minutes based on their lung volume. However, on the other hand, when we look at thermoregulation and how much heat these crocodiles are losing to the water around them, it actually decreases quite dramatically as they get smaller. And so we again have data back from the 70s in this case on crocodiles and how quickly they cooled their bodies when they were submerged in water. And you see that actually at the smallest body sizes, you'd have a diving capacity of less than a minute for the smallest crocodile. And one really cool thing is that if we look at where these two lines cross, that's exactly where we stop seeing marine crocodiles. We no longer see any marine crocodiles below this kind of nick point here, where the amount of energy you lose based on cooling in water crosses the amount of diving capacity you'd have based on your lung volume. And so what I think is going on here is there is actually some very strong constraint on body size imposed by how quickly you cool down, again, in water, just like in mammals. But at a point, that diving capacity meets a trade-off where now you can only support your dives for as long as your lung volume can support them. And so you have kind of this, the minimum of these two lines is the constraint at the different body sizes. So that constraint actually shifts as you get larger. Okay. And so in conclusion here, we again find that there's increases in body size coupled with increases in strength of selection and decreases in variance. And these are following invasions of marine habitats, but not of semi-aquatic habitats. And actually, it's possible that these increases in body size occur even before, based on just the raw data I showed you. It's possible that the ancestors actually have to be larger in order for these species to invade the ocean. If you look at the data here, again, you can't even be smaller than this size, it appears, if you want to be a marine crocodile. So there is some very hard lower bound, just like in mammals. It looks like diving capacity is driving this body size evolution of these groups. At sizes lower than 10 kilograms, thermoregulation imposes a strong minimum size constraint. And above this size, lung volume is possibly also a selection factor. And that might also be driving them towards even larger sizes beyond that strong minimum size constraint of 10 kilograms. Okay, and so some final takeaways here between the two chapters. So there's very strict physiological demands, as I talked about before, in the aquatic medium, and these have caused a variety of adaptations by tetrapods before and during their aquatic evasions. Here I've only talked about two groups, and I've only talked about one type of adaptation. But in most cases, one or more many key innovations are required in order to make these invasions remotely possible. And so in this case, I've shown that they need to have significantly larger sizes in marine mammals and crocodiles to invade the ocean. And these findings suggest that broad-scale patterns of morphological evolution that we see at higher taxonomic levels, like mammalia and crocodiliforms, are often determined more by physiological constraints than by ecological interactions or environmental fluctuations. I just want to thank all the people that helped make this work possible, and the various funding sources that also assisted with that. Thank you all for coming and listening to me. The eoPERCS committee for inviting me, and I'd be happy to take any questions you might
have. Well, thank you, Will. It was an amazing talk. I really enjoyed learning some stuff, and lots of props. I like props. So we already have some questions, but I would like to remember other people that you can send your questions to the questions host, that today is Natasha. So if you like to ask Will any question, just send Natasha a question, or you can raise your hand using the reactions button. First I'm gonna just let you know, Will, I'm gonna copy and paste the questions to the chat as well, so you can read them. So the first question is, does this pattern of oceanic gigantism also extend to extinct or older taxa, like this Mosidia and Metrorhynchids?
I'd love to explore this in other groups. When I started on the mammals, the tree that I had only extended to living things, so I could only look at the living groups. I then did the fossil representatives of those groups, but I didn't look at Desmostylia or the other fossil marine groups like the marine sloths and things. But I think just anecdotally, it looks like those things do also get larger. I don't know about Metrorhynchids, but I'm hoping to expand this as I keep going to other groups. Although I don't want to turn into kind of like a one-trick pony and just look at this question across all these groups. But at the same time, I do think it's a really interesting question, and I imagine that this is such a fundamental constraint that it probably applies to almost every single marine tetrapod group.
Yeah, makes sense. So the next question is, I forgot to say, but the first question was by Sender. The next question is, how would a graph look when plotting only the selection parameter and not the random drift components of the understand neural back process.
Interesting. Yeah. So basically, when you remove that random drift component of the full process, you're just going to get the very smooth curve, which I showed. Hold on 1 second. This is when I'd love to have my second monitor from my normal office. Right. So here, this is basically the lines here are showing what you'd expect based on only that selection component of the OU process, and then the volume around it is the amount of variance that you would expect if you included the random drift component in the process. So you can actually see both of those in here, where that's what you would that's the expectation, that black line of just the selection process, and then there's the randomness around that you'd expect from adding in the random drift component.
Nice. So yeah, another question is, what are some of the assumptions that you made in creating the body size model?
I guess it depends which model you're talking about. If you're talking about these OU models,
I think it was the model you created to predict the marine mammal body sizes.
Ah, yes. Yeah, so it's a very simple model. I think we're talking about this model here. Yeah, it's a very simplistic model intentionally. So, I mean, there's so many other ways you obtain and consume energy in your body. But we wanted to just kind of get at the broad strokes, like the basic way you obtain energy is feeding, and the basic way you lose it is metabolism and in the ocean, heat loss. And so we're trying to get rid of all the kind of minutia that might be associated with this, just to focus on the big things. But yeah, there's certainly a lot of things we're missing here. There's also this is also based on only seals, and it's not based on other groups. I did that because seals were the only group that had data on all three of these things available at the time. It's possible there's more now, but people don't tend to really do these kinds of submerge experiments anymore, probably for good reason. You know, back in the 70s, people were like holding these marine mammals underwater and looking at how quickly they lost water, heat, and like that's terrible. So like, unfortunately, there's not really any experimental stuff going on anymore. And so there's definitely assumptions that like we have enough data and things like that. So yeah, it's riddled with assumptions, but the whole idea is that we're getting rid of all the noise and looking at just at the main signal.
Okay. So yeah, another question by Bethany. So thanks, Will. As you mentioned, it can be hard to distinguish between evolutionary models when there isn't enough data. Do you know whether anyone has tested this in terms of knowing how big a phylogeny is needed, or how much evolutionary time needs to pass to be able to tell the difference, or how strong the selection needs to be?
That is a great series of questions. There was a paper. Now you've got me wanting to look into this. There was a paper at one point on how the OU model is bad, or maybe that wasn't the title. The title was like a cautionary tale of the Ornstein-Uhlenbeck process or something like that. And in that paper, they might have done some sensitivity analyses looking at how big of a tree you need. I don't know if anyone's looked at how much time you need to have passed. You would hope that if it's a biologically sound model, in that things are evolving in this kind of curvature manner and they're evolving faster at the beginning, you would think that you need to get some sort of that asymptotic part of the curve in order to estimate kind of the slope of the curve reliably. So I would think that if things are still kind of in that rapid evolution phase, you might not be able to estimate these parameters reliably. But at the same time, that's also the part of the curve where like it's changing the most. So like the second derivative is the steepest or something. I don't remember calculus. So maybe it would be. So I don't know. Intuition says that like you would need to get to that kind of asymptotic part of the curve in order to really estimate where they're getting. But then that means that it's not a very good model to look at things that are currently evolving, which I think the otters are. Like I would expect otters to keep getting bigger in the near future, near being, millions of years. But at the same time, a lot of the work with crocodiles shows that there's this kind of hard minimum, and they have to get above that in order to even live in the water in the first place. And so a lot of the implications I'm making here is that they're kind of evolving in the water. And that might be true of some of these things that were semi-aquatic for a long time. Like whales were semi-aquatic for a long period of time. And so that's probably when they got larger. And lots of people have looked at this in the fossil record since I've done this work, and show that, they do get larger kind of stepwise as they're semi-aquatic. And so, yeah, I don't know. That was really rambly, but I love the question, and now I kind of want to look into whether anyone's done some sort of sensitivity analysis of that.
Yeah, that's a good question. We can talk more about this in the tea time if Bethany can stay, but just a comment. For crocs, maybe you can see this hard constraint on the lower limit, because we don't, the most significant marine croc group is Thalarasuchia, and we don't see the fossil record many of the transition
Right.
fossils. So we're not seeing what was going on at the beginning of the evolution of the group. Maybe that's why. So another question is now about crocs. Do we have evidence suggesting that extinct marine crocs were ectotherms? If some of them were not, how would this affect your study?
I am not an expert on this, and I know there's been back and forth throughout all of reptilia over whether things were ectotherms. I know there's been evidence both ways, I think, and Pedro might know more about this than I do, but I don't think so. I guess one of the assumptions I make in the model for the crocodiles is that the rate at which they lose heat and produce it in their body is the same as that of modern crocodiles, which are ectothermic. But they also have some funny things that go on for their semi-aquatic lifestyle. So like they have other adaptations, like they keep most of their blood away from their skin, as far as if I recall correctly, so that less of the blood where the heat is able to lose heat to the air around them. And so they do kind of weird metabolic and physiological things to also maintain heat. And so it's not necessarily endothermy or ectothermy. It's probably somewhere in the middle. And it's not really a great, the two terms aren't really great one size fits all to begin with. So I guess what I'm trying to say is they're probably somewhere in the middle. And modern crocs are even probably somewhere in the middle too, but probably closer to ectotherms. It's possible that these marine crocs were closer to endothermy, but probably not completely. And yeah, I don't know if that's that answers your question, but how would it affect my study? I'd say probably not too much. It would probably move things around a little bit in terms of this model, but really this is such a steep relationship that moving it to the left or right a little bit wouldn't change too much.
Yeah, makes sense. Yeah, and I agree. The crocs are something.
Yeah, they're weird. Yeah.
So a question from Shannon is, "Hi Will, great talk. I think I might have asked you this one time at a conference. So I was wondering about a few examples of smaller aquatic tetrapods, for example, sea snakes, marine birds like penguins. Are there factors why these can still be small despite being constraints, as air breathers or endotherms?" etc.
Yeah, it's a great question. And my last chapter, which I didn't have time to talk about of my PhD, was on sea snakes and why they're not big. And all these other groups seem to evolve some other adaptations. And actually, thinking of sea snakes is a great example because they actually seem to do the opposite of what you think a marine adaptation would require, right? They are long and skinny, and that is the worst case scenario for thermoregulation. You are exposing, you have such a high surface area to volume ratio. And so you're losing so much heat to the water around you as a sea snake. And so but actually, if you look at the distribution of sea snakes today, they're all in really warm waters. And so as long as you're in warmer water, you don't have to worry as much about losing heat. And the work I did showed that those sea snakes tended to evolve into warm water. Like they started in warm water. That's where they invaded the ocean. Now that's kind of hard to estimate since we don't have a lot of fossil data, but at least based on the modern things, and if you do kind of some of these weird reconstruction stuff, it indicates that they might have started in warm waters to counteract the thermoregulatory constraints. And so other groups have done other things. I don't know much about marine birds, but I'd love to look into that. But I know that you've got you also, if you're not in the water full time, that also helps. You can, if you're semi-aquatic, it's very different, as you can see in the crocs here, from being marine. You don't have to be above this body size threshold. So I think things like penguins, since they do have this kind of semi-aquatic lifestyle, that certainly helps. And even though they might lose a lot of heat while they're in the water, going back out of the water then helps. And then penguins are almost like um, uh, otters in that they have the really thick feathers, and they basically are insulated, like with otter fur. And so I think there's other adaptations you can do, but there's still always going to be this constraint. It's not necessarily going to be the same body size, depending on if you have other adaptations, and depending on where you live on the planet. But yeah, it's definitely more complicated, I guess, than I'm making out here. There's other things going on, but at least in these two groups, it appears that they do the same. They respond the same way.
So I think this is going to be the last question, also from Sender. So the secondary invasion of mammals, if not for all tetrapods, is much accelerated throughout the Cenozoic. Can this increased rate of invasions be a consequence of the KPG event and the extinction of marine reptiles like mosasaurs, plesiosaurs, and ichthyosaurs?
Yeah, let's see. Let's go back. So yeah, I mean, it does seem like it speeds up towards the Cenozoic. But I mean, there are lots of Mesozoic invasions. All these reptile groups invade. So it's not actually clear to me whether it's faster in the Cenozoic, unless by accelerated you mean like the invasion itself happens faster. One of the things I'm really interested in right now, though, and my next postdoc is going to be working on, is kind of the diversification dynamics of these invasions, and what's driving them to diversify at the invasion point, what causes them to go extinct in terms of biotic, abiotic factors. Why might they not be diversifying as much as they were on land? Those kind of things. I think a lot of that is still kind of unknown, and so I'm hoping to look into a lot of that.
Yeah. Maybe the question was more related to the previous or following graph where it showed the number of species for each group. So, because mammals have more. Yeah.
Oh, yeah. Sorry. Yeah. This is probably the pull of the recent here.
Yeah.
So we just have more fossils from recent times, and so we're probably just getting a better record of mammals. It's also possible that a lot of these mammals are semi-aquatic. I mean, a lot of the reptiles were semi-aquatic as well, but it's possible that if there's more semi-aquatic things, they might have better preservation rates than the fully aquatic things. I don't know. There's probably lots of geological and sedimentological factors involved here that are kind of causing this to inflate. This is probably not a real signal, not completely real. it might be somewhat.
Yeah. Okay, I think that's all for now. I don't see any other question. Yes, so let me take over the screen share. So yeah, thank you, Will, again. It was great talk. Hope like if some of you want to stay for tea time, to ask Will the questions, just feel free to stay. We're gonna just another reminder about the weekly feedback form that you're gonna see in the chat soon. And next week you're gonna be joined by Catherine Hayes from the University of Colorado, Denver. And it's going to be, I believe, the last talk of our special fieldwork seminar. And she's going to be talking about support for students during fieldworks. During tea time, we can keep talking about similar subjects, but we can also talk about other stuff and answer this great question about which is your favorite secondary marine technology. So yeah, we have a short break before tea time. We can go drink some water, go to the bathroom, and we'll be back in 2 minutes. Thank you.